Lesson 3a, deoxyribonucleic acid. DNA is the chemical found in the cell's nucleus, responsible for controlling the cell's activities. The monomer component of DNA is the nucleotide, which is made up of a ribose sugar and phosphate backbone, with a nitrogenous base forming the rungs. These bases could be adenine, guanine, cytosine, or thymine. A single molecule of DNA is composed of two strands of DNA. They are joined by complementary matching base pairs, where adenine hydrogen bonds with thymine, and guanine hydrogen bonds with cytosine. The DNA then twists into a familiar double helix or twisted ladder form. DNA further folds around proteins called histones to make thicker strands called chromatin. This chromatin is the form that DNA is usually found within the nucleus. This form of DNA allows for easy copying into identical strands, a process called DNA replication, or also for easy copying into RNA so that proteins can be made. This process is called DNA transcription. DNA is often illustrated as a more compact and tightly wrapped structure called chromosomes, which we often see as a figure X. DNA is very fragile, and it forms this dense package to ensure that it will survive the process of cell division, called mitosis. In fact, the X shape of the chromosomes is not a single molecule of DNA, but rather two identical molecules called sister chromatids, made through a process called DNA replication. These sister chromatid arms are held together by a protein called the centromere, until the time the cell goes through mitosis, where it divides into two. This duplication of the DNA is necessary, so that when the cell divides, the two cells formed each have a full set of DNA molecules. Each normal body cell found in humans has 46 chromosomes, or 23 homologous pairs of matching chromosomes. When a cell has a full set of chromosomes, it is said to have a diploid number of chromosomes. The full complement of DNA can be seen in a diagram of a cell's chromosomes, called a karyotype. In this karyotype, the 46 chromosomes appear as bars, so this cell has yet to replicate its DNA. In this karyotype, each chromosome appears as an X, ready for mitosis. This cell has replicated its DNA in preparation for cell division.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.